Hello everyone. Today's topic is chromosome structure and their function. Let's see chromosome. The chromosomes are the rod-shaped dark-stained bodies are seen during metaphase stage of mitosis when the cells are stained with suitable basic dye or viewed under a light microscope. So chromosomes are first described by Strasburger in 1875. The term chromosome, however, was first used by Walder in 1888. Let's see the chromosome history. So chromosomes first described by Strasburger, which was seen in that Walder used the term. So in 1842, Carl Wilhelm, which was a Swiss botanist, discovered a structure which later known as chromosomes. In 1882, Wilhelm Hofmeister denoted the term as chromosomes. In 1883, Wilhelm Roig speculated chromosomes are the carrier of genetic information. So Walton Sutter and Theodore Bauer in 1902 suggest that chromosomes are the physical carriers of the genes in the eukaryotic cells. The compactness of chromosomes plays a vital role in the helping to organize genetic material during cell division and enabling to its fit inside structures such as nucleus of the cell. So the chromosomes they were given the name chromosome, which means chroma means color plus soma is equal to body due to their marked affinity for basic dyes. The chromosomes are clearly visible as a distinct bodies during the stages of cell division only. Their number can be counted with relative ease only during mitotic phase. The interphase nucleus, on the other hand, does not exhibit any structure resembling chromosomes. Under the light microscopic studies by Bower in 1890 and subsequently by other workers clearly established that their integrity of metaphase chromosomes is maintained through the interphase, that they do not disintegrate during interphase. Let's see the chromosome number. Each species has a definite and generally a constant somatic and gametic chromosome number. So the somatic chromosome number is the number of chromosomes found in the somatic, more specifically meristematic tissues of a species and represented by 2n. Ordinarily, somatic cells contains two copies of each chromosome. The two copies of each the two copies of chromosomes are ordinarily identical in morphology, gene content, and gene order. They were known as homologous chromosomes. Gametic chromosome number is precisely one half of the somatic number and is represented by n. It is denoted by the number of chromosomes found in the gametes of a species. It is important to note that gametes of a species ordinarily contain one and only one copy of each of the different chromosomes of that species. Thus, in any individual, one homologue of each chromosome comes from its male parents, while other contribute contributed by its female parent. Let's take the chromosome size. The size of chromosome shows a remarkable variation depending upon the stage of cell division. Chromosomes are the longest and the thinnest during interphase, so that they are not even visible under light microscope. With the onset of prophase, there is a progressive decrease in their length, accompanied with an increase in thickness, that is diameter. So the chromosomes are the smallest during anaphase, but chromosomes are the most easily observed and studied during mitotic metaphase, when they are very thick, quite short, and well spread in the cell. Therefore, chromosome measurements are generally taken during mitotic phase only. The size of mitotic metaphase chromosomes of animal and plant species generally varies between 0.5 mu to mu and 32 mu meter in length, and between 0.2 and 3.0 mu in a diameter. The longest metaphase chromosomes are found in trillium. The longest chromosome, that is 32 micro in the microbial present. The gain chromosomes found in a diphtheria. Therefore, gain chromosomes are easily visible even in the interphase nucleus. Let's let's see let's see about chromosome structure. So the chromosome structure contains many organelles, many organelles such as, so each cell has pair of each kind of chromosomes known as homologous chromosomes. So the structure of chromosomes can be seen during cell division. Each chromosome contains hundreds and thousands of genes that are precisely coded for several proteins in the cell. So the chromosomes are made up of chromatin, which contains a single molecule of DNA and its associated protein. The first, let's say the first part, first uh, The following the some features will be present for each of the chromosome part. So let's to first one is discussed with chromatid. So each each metaphase chromosome appears to be longitudinally divided into two identical parts, each of which is known as chromatid. So each chromosome has two symmetrical structures called chromatids or sister chromatids, which is variable in mitotic phase. The chromatids of a chromosome appear to be joined or fused together at a point called centromere. So each chromatid contains a single DNA molecule at anaphase of mitotic cell division. Sister chromatids separate and migrate into opposite poles. In contrast, that chromatids of homologous chromosomes are referred as non-sister chromatids. Next is centromere and kinetochore. Sister chromatids are jointed by centromere. Spindle fibers during cell divisions are attached to the centromere. So the number and position of the centromere differs in the different chromosomes. The centromere is a primary constriction. Centromeres divide the chromosome into two parts, where the short one is known as P-arm and the longer arm is known as Q-arm. The centromere contains disc-shaped kinetochore, which is specific DNA sequence with special proteins bound to them. The kinetochore provides a center for polymerization for tubulin and assembly of microtubules. Next, next one is chromosomes are divided into two transversely parts by the centromeres. These parts are called arms. As a result, centromeres are the first parts of the chromosomes to be seen towards the opposite poles during anaphase. Next one is secondary constrictions. Secondary constrictions, these other than centromere, uh, chromosomes possess secondary constrictions, where secondary constrictions are be identified from centromeres at anaphase because there is bending only a centromere primer constriction. The chromosome region lying between secondary constriction and the nearest telomere is known as satellite. Generally, secondary constrictions are located in the short arm of chromosome near one end. Secondary constrictions which contain gene to form nucleoli are known as nuclear organisms. Chromosomes having secondary constrictions are called satellite chromosomes or SAT chromosomes. Nucleus is always associated with the secondary chromosomes or satellite chromosomes. Next, next term is telomere. Terminal part of a chromosome is known as telomere, which is end of the chromosome. The telomere are polar, which prevents the fusion of chromosomal segments. Telomeres are highly stable, and telomeres of different chromosomes do not fuse or unite. In case the end of telomeres consists of a folded DNA strand, as a result, that these ends are stable and resistant to exonuclease digestion. Next is chromatin. Chromatin is made up of chromosome is made up of chromatin. The chromatin is made up of DNA, RNA, and proteins. At interface, chromosomes are visible at these thin chromatin fibers present in the nucleoplasm. During cell division, the chromosome fibers condense and chromosomes are visible with distinct features. The darkly stained condensed region of chromatin is known as heterochromatin. It contains tightly packed DNA which are genetically inactive. The lightly stained diffuse region of a chromatin is known as euchromatin. It contains genetically active and loosely packed DNA. At prophase, the chromosomal material is visible as thin filaments known as chromonimata. At interface, bead-like structures are visible, which are accumulation for chromatin material called chromomere. Chromatin with chromomere looks like a neck-like with beads. During early and mid-prophase stages, there are three deeply staining structures in the chromosome that are chromomeres, centromeric region, and knobs. The chromomeres may not be present to may not represent true heterochromatin. Centromere regions invariably contains heterochromatin. The knobs are the spherical structures serves uh, usually several times the diameter of a concerned chromosomes. They are present in on the certain chromosomes of some species. Example like maize. Let's see uh, difference between the heterochromatin and euchromatin. So they, as further we discuss, heterochromatin are darkly stained regions, while the euchromatin are lightly stained structures. This contains few inactive genes. It contains a lot of active genes in the euchromatin. Usually found near centromere and telomere. It's heterochromatin usually found near centromere and telomere, while euchromatin found in the middle of the chromosome between centromere and telomere. The heterochromatin is again further divided into constituent facultative, but euchromatin is only one single type. But in heterochromatin, replication will be slow process. It takes some time. The euchromatin, it is a normal replication. But in heterochromatin, usually no active part in the transcription. But euchromatin plays a vital role in the transcription. Heterochromatin contains 30 nanometer fiber, where euchromatin is 3 to 8 nanometer fiber only. But heterochromatin covers small region of the chromosome, where euchromatin covers larger region of the chromosome. So the let's see the heterochromatin uh, types, that is constituent faculty too. The constituent heterochromatin remains permanently in the heterochromatic stage, that is it does not revert into euchromatic stage, that is in the centromere regions. In contrast, facultative heterochromatin is essential euchromatin that has undergo heterochromatization, that is conversion into heterochromatin. Heterochromatization may involve a segment of chromosomes, a whole chromosome, or one haploid set of chromosomes. In general, heterochromatin is found in the centromeric and telomeric regions, but the other regions are also be heterochromatin. Heterochromatic chromosomes and chromosomes region in the chromosome region ordinarily replicate later than to go for uh, euchromatin regions. Next part in the chromosome is matrix. The mass of achromatic material which surrounds the chromonimeta is called matrix. The matrix is enclosed in a sheath which is known as pellicule. Both matrix and pellicule are non-genetic materials and appear only at metaphase when the nucleolysis disappear. Next is karyotype and ideogram. The general morphology, that is size of chromosomes, position of centromere, presence of secondary construction, and size of satellite bodies of a somatic chromosomal complement of an individual consists are known as karyotype. It can be defined as the characteristic feature by which a set of chromosomes of a species is identified. Generally, karyotype is represented by arranging the chromosomes in descending order of a size, keeping their centromere in the same line. Thus, the largest chromosome is placed on extreme left and the shortest chromosome on extreme right. The karyotype of a species can be represented diagrammatically, showing all the morphological features of chromosomes. Such a diagram is known as ideogram. Next, we will see structural organization of chromatin. So, the chromatin contains of DNA and associated proteins. As further I told, DNA is packed in a highly organized manner in the chromosome. Next, first we'll see nucleosomes are the basic unit of chromosomes. So it is 10 nanometer in the diameter, where the DNA is packing is facilitated by proteins called histones. DNA is wound around histone proteins to form a nucleosome. There are 5 types of histone proteins in the eukaryotic chromosomes, namely H1, H2A, H2B, H3, and H4. Histones are positively charged due to presence of amino acid with basic side chains and is associated with negatively charged DNA due to presence of phosphate groups. Histone proteins plays an important role in the gene regulation. Nucleosomes are made up of 146 base pairs of DNA coiled around a core of 8 histone molecules, where 2 molecules of 4 histone proteins. Nucleosomes prevent DNA from getting tangled. Next is the DNA, the linker DNA in the fifth histone, which is H1, packed adjacent nucleosomes to a 30 nanometer compact chromatin fiber. These fibers form a large coiled loop held together by non-histone proteins, where actin, alpha, and beta tubulin myosin called scaffolding proteins to form extended chromatin, which is 300 nanometer in diameter. Chromatin further condenses with the help of proteins known as condensin. It binds to DNA and wraps in the coiled loops, and we get the combined chromosome. Next, we'll see type of chromosomes. So the type of chromosomes mainly based on the centromere were present. First one is based on number of centromeres present. That is, first one is monocentric. So it's having only one centromere. Next is holocentric, having diffuse centromere and microtubules are attached along with the length of chromosome. Next one is eccentric. The chromosomes may break and fuse together to form a chromosome without a centromere, so it cannot attach the mitotic spindle. Next one is dicentric. Chromosome aberration, where chromosome break and fuse together with two centromeres. So they also unstable as their two centromeres tend to migrate to opposite poles resulting in the fragmentation. So based on the position of centromere, there are five types, that is telocentric, where is a rod-like chromosome with centromere present on the proximal end. So no p arm, no, number of p arms, this is short arm present. Telomere chromosomes are not found in humans. Next is acrocentric. There are rod-like centromeres present at one end, giving rise to only very short arm and exceptionally long arm. Next one is submetacentric. L-shaped or J-shaped with centromeres near the center of the chromosome, giving rise to two unequal arms. Where metacentric, V-shaped chromosome with chromosome in the middle, giving rise to two equal arms. Next one is holocentric. Last one is holocentric, where the chromosomes having the diffuse centromeres. Next one is gain chromosomes. There are two gain chromosomes, which are polytene chromosomes and Lamprecht chromosomes. First we discuss polytene chromosomes. So Balbani first discovered a structure in the nuclei of secretory glands of midges. So Painter, Heids and Baer rediscovered them as salivary glands of Drosophila and recognized them as chromosomes, also known as salivary gland chromosomes. They are called polytene by Collard due to the presence of many chromonimata in them. These are present in some of cells of large of diphtheria insects also. These are very large due to presence of high DNA content. The Walton chromosomes of Drosophila salivary glands has 1000 DNA molecules, uh, it has 1600 DNA molecules in each polytene chromosome. There is a series of alternating dark and clear bands called interband. Chromosome puffs or Balbini rings are present, which are swelling of bands due to DNA unfolding into open loops. So these are the regions of the intense transcription or mRNA formation. Next is Lamprecht chromosome, first discovered in the oocytes of salamander. This name is given due to its resemblance with a brush that is used for cleaning lamp glasses, chimneys, etc. They occur at the diploidal stage of oocytes in, uh, in vertebrates and in oocytes in vertebrates and invertebrates. So Lamprecht chromosomes are also found in spermatocytes of many animals and also found in gain nucleus of an algae. They are present as a bivalent with four chromatids. So lateral loops of DNA are always symmetrical and found due to intense RNA synthesis. In the oocytes of salamander, there are 10,000 loops present per haploid set of chromosomes. The centromere does not bear any loops. If see the replication of the chromosome, the mode of replication of eukaryotic chromosomes like that prokaryotic chromosome, which are semi-conservative type. So this was first demonstrated in 1956 by J.H. Taylor and co-workers in the Vicia paper. Chromosome replication is more complex than DNA replication. Next, next we will see chromosome banding. The banding in the 1970, spectacular technique has been developed for identifying any chromosome, that is chromosome banding. So the banding pattern each chromosome is fixed and usually consists in a taxon. So it was discovered that chromosomes stained with fluorochrome correctnine or its derivative fluorescence in alternative bright and dark bands when examined under ultraviolet microscope. So different types of bands are obtained by using different types of techniques, and these are made visible through low and high intensity regions under the fluorescent microscope, or say 7 of different stained bands under the optical microscope. So that is easy to identify a chromosome with the binding patterns of human beings and identify by the fingerprints. So these are the types of bands of techniques, which is Q band, that is obtained by the fluorochrome correctnine, quinacrine. Next G band obtained by using the JSMA technique. Next one is R banding, which is reverse to Q band, which trained with JSMA after heating at 87 degrees Celsius. Next is C band, demonstrated by the denaturation reassociation technique, showing the constitutive heterochromatin. Next one is E band, obtained by enzymatic digestion. Next one is CT band, where centromeric and telomeric bands after barium hydroxide and stains all staining in the mammals. Next one is N band at the nucleolus organism regions in the mammals. O band with fluorescent staining, intercalary and heterochromatin bands made in the plant chromosomes. Next, next we will say functions of chromosome. The main function of chromosome is to carry the genetic material, which means one generation to another generation. They play a vital role in act as guidance for the guidance force in the growth, reproduction, repair, and regeneration process. So that is important for their survival. Chromosomes protect the DNA from getting tangled and damaged. Histone and non-histone proteins help in the regulation of gene expression. Spindle fibers attached to centromere help in the movement of chromosome during the cell division. So here are the functions of the chromosomes. So today's topic is ended with the chromosomes structure and functions. Thank you.